Hello everyone. Welcome to today's live broadcast, Powerful Tools for In Vitro Fluorescent Imaging in Cancer Research. I'm Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by LabRoots.com, a leading social media site for science professionals, and sponsored by ImmunoChemistry Technologies. ImmunoChemistry Technologies provides quality laboratory services and research reagents. As a contract service laboratory, ICT specializes in custom immunoassay development, manufacturing, conjugation, lyophilization, and consulting services. ICT is focused on ELISA development and has the knowledge to develop reliable, sensitive, and specific tests. ICT offers a line of ELISA dilutants, including coat buffers, conjugate stablers, and sample dilutants designed to reduce backgrounds, increase specific signal, and improve the stability and shelf life of the ELISA. ICT also offers a line of fluorescent reagents to measure caspase activity, apoptosis, and cell death in vitro. Before we start, there are a few instructions for this presentation. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many of them as we can. If we don't have time today, we'll definitely follow up. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right, or use the Q&A button. We'll try to make sure we resolve any issues. Also, we want to let the audience know that this presentation has been approved for continuing educational credits. If you want to obtain the credits, please click on the Get Your Free CE/CME Credits button located in the lower left of your screen. This will take you to a page listing all of our presentations and speakers. Please select the appropriate CE/CME button under your presentation and follow the instructions to claim your certificate. Now let's get right to today's presenter. We are proud to welcome Jackie Carville. Jackie Carville is the Director of Marketing at ImmunoChemistry Technologies, where she focuses on providing comprehensive, user-friendly content to assist life science researchers in optimizing their project pipelines. She obtained her undergraduate education at University of Wisconsin-Madison, studying genetics and life science communication. For more information on Jackie's experience, please click on her name. I'll now turn the presentation over to Jackie.
Great, thank you so much, Brenda. Hi everyone, thanks again for joining me for today's presentation. As Brenda mentioned, I'm Jackie Carville and I'm the Marketing Director at ImmunoChemistry Technologies. Today I'm going to be going over a variety of powerful tools for in vitro fluorescent imaging, how these tools work, and their applications for cancer researchers. Just as a little FYI, when we're getting started, there were a few import issues with the slide deck, so there might be a little off formatting you'll see, but don't worry, we'll have the slides available for your download if you would like clarification and read these slides as well. But to get started here today, I'd like to give a little background about ImmunoChemistry Technologies. So here at ICT, we offer quality biomedical research reagents, including unique apoptosis assays and proprietary immunoassay reagents. We serve researchers all over the globe and are dedicated to helping our customers achieve results. We were founded in 1994 in Bloomington, Minnesota, which is located in the greater Twin Cities area, just south of Minneapolis that we have the picture of here. Originally, ICT was a contract laboratory focusing on custom immunoassay development work and ELISA reagents, but we have expanded and diversified our product lines over time. So today, we're no longer just a contract laboratory for protein chemistry. Now we're your trusted supplier for apoptosis assays, ELISA detection reagents, and custom ELISA development and other protein chemistry-related services. Our customers use our products to study human and animal biology and to investigate treatments for cancer, degenerative diseases, and other conditions affecting both humans and animals. So as a note, all of our products and services are designated for research use only. So I'd like to talk a little bit first to get started today about our ELISA Solutions products. These products are designed to help you build a better assay. Our products address the issues that typically occur during ELISA development, which involve specificity, sensitivity, reproducibility, and shelf life of the product. Now, our ICT coating buffers, blockers, sample and assay diluents, conjugate stabilizers, and wash buffers all work together to minimize the buildup of unwanted proteins and to generate a very clean signal. In this area, we do focus on providing several key ways to optimize your ELISA assays. So first, you're going to want to minimize your background and enhance your signal-to-noise ratios for improved assay sensitivity by promoting a strong signal while minimizing nonspecific binding and background noise. Next, you can also improve your precision and reduce plate-to-plate variability through the use of our stabilizing buffers, which allow you to prepare plates in large batches to use over time, therefore increasing your consistency and providing that improved plate-to-plate precision over extensive storage periods. Additionally, you can increase your assay reproducibility by purchasing consistently through a reliable source. Also promote a higher specific signal by minimizing non-specific binding through specialized blocking and stabilizer formulations of our products. You can also stabilize your protein conjugates through the use of alkaline phosphatase and horseradish peroxidase conjugate stabilizers. And finally, increase your shelf life of your prepared microtiter plates by correctly stabilizing adsorbed proteins with reliable coating and blocking buffers, resulting in plates that can be stored for several months or even years under the proper conditions. So to summarize all that information, ELISA development really is a core specialty of ours, and we want to work with you to help optimize all your immunoassay work. So as I mentioned, in addition to our ELISA services, we also offer a wide range of various custom contract services as well. So with regards to the ELISA services, if you're like most scientists, you really want to focus on your field of study and don't necessarily have either the background or the time in order to develop a rugged immunoassay test yourself. So pre-developed ELISAs and other immunoassays may be easy to run, but the development itself requires years of knowledge of immunology and protein chemistry to really understand antibody specificity and binding interactions. And that's where we come in at ICT. So our scientists are experts with many, many years of experience in immunoassay development and can also perform optimization and testing services. Now, outside of immunoassay development, we do also provide custom protein lyophilization services. So if you need a reagent vialed or freeze-dried, we can reliably prepare that for you. Most lyophilization projects, we can complete those in about a week, so there's a pretty quick turnaround for you on those types of projects. So with that said, I'm going to go ahead and move on to our fluorescent probe product lines, where we'll be focused for the rest of the presentation today. Now, our fluorescent cell-based assays are for apoptosis detection, which is a form of cell suicide which is mediated by a cascade of proteolytic enzymes known as cysteine proteases, or caspases. So caspase assays, live dead stains, and mitochondrial permeability assays are all essential in the investigation of cellular apoptosis and cell death. So what I'll be going through here in today's presentation are these 5 different fluorescent probe product lines. So first we'll go through Flicka, which is our product line for the detection of active caspases, Magic Red for the detection of caspases and cathepsins in real time, MightOPT to assess mitochondrial health, Cytotoxicity Tests, and Fliss for serine protease detection. So I'm going to start here today with one of our more popular products, which is our Flicka kits. Now, Flicka stands for Fluorescent Labeled Inhibitor of Caspases and measures cell death by detecting intracellular caspase activity and membrane permeability in whole cells. We do offer these kits in 3 different color options for green, red, and far red. These kits, as well as many of our other assay kits we'll discuss today, are offered in different sizes with varying numbers of tests, with trial size options available. So as you're assessing your particular experiment and looking to determine what the number of tests are, we do define a test as a 300 microliter aliquot of cells that are grown at no more than 1 × 10⁶ cells per milliliter. So just something to keep in mind as you're thinking about your own experiments as we go through some of these assays today. So in terms of cancer research, there are several advantages to using Flicka. And first, Flicka is the most accurate and sensitive method of apoptosis detection, as it measures the actual intracellular process of apoptosis via the caspase activity, instead of measuring a side effect of the process. So second, it's the only method that can detect cells in early apoptosis and differentiate them from cells that are in late apoptosis. So you really get more specific information out of these types of assays. So here that also means that you can see how your cells are responding to treatment and determine whether they're apoptotic, necrotic, or unaffected cells. And finally, we do have a few different types of Flicka kits available. So depending on your cancer research goals, we do have different options to suit your needs. So to assess apoptosis in general and look at a variety of caspase activity, we do have Flicka polycaspase assay kits available. If you'd like to target a specific caspase, we have specialized Flicka kits for caspases 1, 2, 3, and 7, 6, 8, 9, 10, or 13. So how does Flicka work? Our Flicka probes, as you can see on the little diagram on the left of the screen, are comprised of a caspase affinity peptide sequence, a fluoromethyl ketone moiety that facilitates an irreversible binding event with the targeted active caspase enzyme, and a fluorescent tag reporter. So I'll kind of open up on the side here the different steps of the process. Flicka is a cell-permeable direct probe, which means that the cell does not require lysing in order for this assay to work. It also does not require permeabilization, additional reagents, or antibodies for detection. So to use Flicka, you simply add the non-cytotoxic Flicka probe to the cell culture media. Once Flicka has diffused into the cells, it will interact with the enzymatic reactive center of a targeted active caspase and form an irreversible covalent bond, marking the cell with its fluorescent signal. There is no interference from inactive forms of the enzyme during this process. Now, unbound Flicka reagent is washed away in the protocol, and the remaining fluorescent signal is a direct measure of the caspase activity present at the time of Flicka addition. You can choose to visualize your cells with a fluorescence microscope, or you can quantify the fluorescence intensity using a fluorescence plate reader or flow cytometer. Green fluorescent FAM Flicka probes excite at 488 and emit at 530. The red fluorescent SR Flicka probes excite at 565 and emit at 595. And finally, the far red fluorescent Flicka 660 probes excite at 660 and emit at 690. So now that we know how the process works, I'd like to now take a look at some different example data results, starting with some flow cytometer data here. And what you're looking at here are results from 3 different Flicka products. So first is our 660 Flicka polycaspase product, which is a far red fluorescent probe, and that's the graph you're seeing all the way on the left there. And what you're looking at there are Jurkat cells that are treated with stereosporin. This is a chemical known to induce apoptosis, so it makes for an excellent positive control. The red line here are our untreated cells, which in this case we'll classify as our control population, and they are non-induced, so these cells are non-apoptotic. The black line on the flip side will be our experimental population, so these are the induced cells and the apoptotic cell line. And as you can see across all of these products, there's a strong shift in terms of fluorescence intensity among the treated cells. In terms of these other graphs, the middle graph is showing a green fluorescence Flicka kit, and the end graph is showing another far red Flicka kit. However, this is measuring pyroptosis instead of apoptosis. On this slide, we have some sample plate reader results. Here again, we're looking at the Jurkat cells that are treated with stereosporin and were then read on the plate reader with excitation and emission at 550 and 590. Here we were staining with our polycaspase detection kit. So again, those Flicka polycaspase kits look at caspase activity more in general as opposed to looking at one specific caspase. And again, in our induced cell population, we can see that there is a strong increase in fluorescent signal. So finally, we do have here some sample fluorescent microscopy results. And on the left, in that first image there, you can see some THP1 cells that are duly stained with our FAM Flicka polycaspase assay kit and hooks to stain the DNA in the cells blue. And if we look at this image, we can see three different cells that are pictured there. The one in the middle is the one we believe to be apoptotic, as we can see green fluorescence for the caspase activity. We also see in that middle cell some bright blue spots in the cell, indicating that the cell is beginning to die as the DNA is fragmenting. The cell in the upper left corner is necrotic, as we see no green and we do see some scattered blue. Then the cell down at the bottom right there, we consider to be live and non-apoptotic, as it lacks any green fluorescence, and it has one really bright concentrated blue spot that does represent the DNA. So the second image on this slide on the right is of colorectal epithelial cells that were infected with salmonella. And here the authors of this particular paper used a salmonella strain that is constitutively expressing M. cherry. So the salmonella appears red in this particular image here. The authors then used our FAM Flicka caspase-1 assay to look for the activation of pyroptosis. So that will stain green then. And you can see that these cells in the middle that are heavily affected with salmonella also do have a strong activation of caspase-1, indicating that the salmonella is that the salmonella infection here is causing these cells to undergo pyroptosis. So with that said, I'd like to now go ahead and move on to our line of magic red assays. So our magic red reagents are cell-permeant fluorescent substrate probes that are used to quantitate both caspase or cathepsin activity in cultured cells. Now, these magic red kits do allow you to monitor your caspase or cathepsin activity in real time, so you can watch your cultured cells undergo apoptosis. These kits are not ELISAs and they do not use antibodies, but instead they are cell-permeant substrates specifically targeted by the active enzyme. And we do offer a few different types, again, of these magic red kits, including those that assess activity of the cathepsins B, K, or L, as well as assessing the activity of caspases 3 and 7. So in terms of cancer research, elevated cathepsin enzyme activity in the serum or the extracellular matrix often signifies a number of different pathological conditions. So cathepsin-mediated diseases do include Alzheimer's, numerous types of cancer, autoimmune diseases like arthritis, and the accelerated breakdown of bone structure as seen with some different forms of osteoporosis. Specifically, upregulated cathepsins B and L activity have been linked to several types of cancer, including cancer of the colon, pancreas, ovaries, breast, lungs, and skin. Upregulation of cathepsin K has been shown in lung tumors. This increased cathepsin K activity has also been shown to be linked to some degenerative bone diseases, including some osteoporosis and postmenopausal osteoporosis. So in general, assessment of elevated levels of extracellular cathepsins has proven to be a very important prognostic indicator of cancer chemotherapy outcomes. So magic red works when the target substrate peptide sequence is linked to a red fluorophore. And magic red, which is also known as crescel violet, fluoresces red once the substrate is cleaved by the specific enzyme. And in this case, that specific enzyme would be either your caspases 3 and 7 or your cathepsins BK or L, whichever one you're interested in. If it is cleaved by an active protease, the released magic red fluorophore will fluoresce, stay inside the cell, and often will aggregate within lysosomes. So the cathepsin enzymes are lysosomal, the caspases are not. And we'll see that in some of the example data as well. So as this protease activity progresses and more substrate is cleaved, the red fluorescent signal increases. So you can actually watch this color develop over time. The reactions are fast, starting within 15 minutes, so you can watch the fluorescence develop over several hours. The typical incubation time for this assay is about 15 to 20 minutes. So you can watch the cells then through a microscope and take pictures of the progress, or you can choose to read the total fluorescence of the sample with a fluorescence plate reader. And magic red does excite at 540 to 590 and emits at greater than 610. So now we'll go through a few examples of magic red results here. So on this slide, we can see an example of what a magic red time lapse might look like. So here, rat fibroblasts were used with the caspase, with the magic red caspase 3 and 7 kit here. So the red fluorescence obviously becomes brighter as caspase activity and apoptosis progressed over the time period of the experiment. And we do have the nice images here that depict that, but if you're interested in magic red and this type of time-lapse progression, we do have a nice video of this same data on our website, so you can watch those cells get brighter and brighter as they undergo apoptosis. But here's another good example of a positive cathepsin B magic red assay in THP1 cells. And so you can see on the left there, where it's fluorescing red, you can see that the cathepsin B is concentrating in the lysosomes of the cells. And finally, this image here is a nice example of dual staining of camptothecin-induced MCF7 breast cancer cells. Here we used both magic red and hooks to stain. So the experimental cells in this case were stained with magic red for about 30 minutes and then washed twice in PBS. And then they were also stained following that with hooks for about 10 minutes. And so again, the magic red staining here for the cathepsin activity and the hooks to stain the DNA in the cells. So you can see the apoptotic cells bearing the orange lysosomal bodies with less intense blue nuclei. The non-apoptotic cells still do have those bright blue nuclei, and they don't have any of that orange lysosomal staining here. So in this particular experiment, it was determined that the treatment that was being applied to these breast cancer cells was indeed killing the cells. So now I'm going to move on to our MightOPT product line and our kits here, which allow you to quantitate mitochondrial functionality in apoptosis. So detection of the mitochondrial permeability transition event provides an early indication of the initiation of cellular apoptosis. So this process is typically defined as a collapse in the electrochemical gradient across the mitochondrial membrane. So these kits will detect a change in mitochondrial membrane potential. These kits are fast, with incubation typically only being 15 to 20 minutes. And we do offer these kits in 3 different forms, this JC1, TMRE, and TMRM kits. And I'll talk through over the next few slides what the differences between those types of kits are. But first, I'd like to talk briefly just about why looking at mitochondrial health is important in terms of different disease research. It's definitely important because the depolarization of this inner mitochondrial membrane can often lead to an opening of the mitochondrial permeability transition pore. This results in the leakage of intermembrane proteins that facilitate apoptosis. Because this mitochondrial dysfunction has been closely tied to such neurodegenerative diseases such as Alzheimer's, Parkinson's, lateral sclerosis, mitochondria do remain an important organelle of study for a variety of different diseases in terms of apoptosis analysis. So as I mentioned, we do have some different types of MightOPT assays which work in slightly different ways. So the first, I'm going to go through how our MightOPT JC1 assay works. And these JC1 assays were our original MightOPT reagents. And they're based off of a fluorescent cationic dye, which penetrates cells and enters the healthy negatively charged mitochondria. Here it aggregates and then fluoresces orange or red. And as the mitochondrial membrane potential collapses, MightOPT JC1 is diffused throughout the cell. And once dispersed, the reagent assumes a monomeric form and then fluoresces green. So this change in fluorescence can be read with a fluorescence plate reader, flow cytometer, or a fluorescence microscope. MightOPT JC1 excites at 488 and emits at 590. So with these sample MightOPT JC1 results, Jurkat cells were treated with cephalosporin for 2 hours to induce apoptosis, and other cells were treated with DMSO as the negative control. These cells were then stained with the MightOPT JC1 reagent for about 20 minutes and then washed. And if we look at this data image, we can see that the two cells on the left are two healthy cells. So you can see the mitochondria there with the polarized inner membranes fluorescing red. On the right of this image, we see 3 cells that are apoptotic cells. And in contrast to the 2 cells on the left, here we can see that the mitochondria are at various stages of permeability so that they do exhibit reduced red fluorescence and increased green fluorescence as the MightOPT JC1 reagent disperses throughout the cell. So our other types of MightOPT assays are TMRM and TMRE assays. And these differ from JC1 in that they are based on different cationic fluorescent redistribution dyes. So the TMRM is based on tetramethylrhodamine methyl ester, whereas the TMRE is based on tetramethylrhodamine ethyl ester. So just 2 different dyes that are used there. Similar to what we discussed with the JC1 kits, after the reagent is added, it does enter the negatively charged mitochondria, it accumulates, and then it fluoresces red. And when the mitochondrial membrane collapses in apoptotic cells, the TMRE or TMRM dyes can no longer accumulate within those mitochondria, and then the dye disperses throughout the cells. The overall cellular fluorescence levels drop dramatically, and this event can be easily detected by monitoring the amount of emitted fluorescence, which can be done through a flow cytometer, plate reader, or fluorescence microscope. So the MightOPT TMRE and TMRM excite optimally at 549 and 548 respectively, and they do yield excellent results using a 488 laser. So optimal emission for these are 574 and 573 respectively as well. So here we're looking at some sample results, and you can really see the difference between the healthy and apoptotic cells, with those depolarized apoptotic cells exhibiting much lower levels of fluorescence. And so you can really see here too, not only the difference between the healthy and dying cells in this data example, but just the difference between the readouts of the TMRE and the TMRM kits versus what we looked at as the readouts for the JC1 kits. So I'm going to shift gears again here to a different product line of ours, and I'll introduce our cytotoxicity assays. So at ICT, we have now developed 2 different assays to measure cytotoxicity. So if you're looking to measure necrotic and membrane compromised cells, you can opt to use our basic cytotoxicity test. However, if you're looking to further distinguish apoptosis from necrosis, leading to a more accurate assessment of cytolytic activity, we do offer a total cytotoxicity test. So with this total cytotoxicity test, at least 10% more cytolytic activity is often measured when apoptotic cells are also identified with this test kit. So these kits provide a direct measurement of cytotoxicity rather than using an indirect indicator, such as the release of LDH or ATP enzymes. And there's no need to lyse your cells for these cytotoxicity studies. Since these assays do not use any radioisotopes in them, such as chromium-51, they are much safer and less expensive, much more cost-effective than other types of assays for cytotoxicity. So there's also no waiting with these kits. You can directly measure cytolytic activity without having to wait for enzyme release. So in terms of cancer research, cytolytic activity is a very important process for eliminating intracellular pathogens and cancer cells. So this process is accomplished through various immune effector mechanisms, including natural killer leukocytes. Natural killer activity is facilitated by non-specifically lysing infected targets through the use of natural killer receptors, so recognizing IgG bound to specific antigens on the target cell's surface. Natural killer cells may also induce apoptosis in target cells. The activity of natural killer cells and their effect on target cells is frequently studied in immunomodulation experiments. So first I'm going to go ahead and run through how our basic cytotoxicity tests work using this nice little diagram we have. First, you purify your target cells and you stain these with CFSE, which is a green membrane stain, to distinguish them from unstained effector cells during flow cytometry analysis. Next, you'll go ahead and add your effector cells or toxic agent to the target cells. And these effector cells or toxic agent will act on these target cells in this step. Next, you can go ahead and add the red live dead stain, which is 7-AAD. And this stain will enter cells with compromised membranes in necrosis and late apoptosis and bind to DNA. It will not enter any cells with intact membranes. So the final step of this process here is to run controls and then analyze using flow cytometry to quantify two populations of target cells in each sample, both the live target cells and the necrotic cells. So the percentage of cytotoxicity can then be calculated by counting the number of dead target cells, which are stained both green and red, and then dividing that by the total number of live target cells, which are the all green cells. So to expand on the capabilities of this basic cytotoxicity test, now we're going to take a look at how the total cytotoxicity test works. And the first two steps of this kit are the same, which is the purifying the target cells, staining with the CFSE, and then adding the effector cells. Where this protocol starts to differ is that in the next step, you'll go ahead and add the orange-red SR Flicka apoptosis detection reagent. So this will go ahead and enter the cells and then bind to active caspases. If the cells are not apoptotic, it will go ahead and wash out of the cells. Then at the next step, we would add our live dead stain, the 7-AAD again, which, like in the basic kit, is going to enter the cells with intact membranes, or with compromised membranes, excuse me, in necrosis and late apoptotic cells, and go ahead and bind to that DNA. The 7-AAD stain is not going to enter any cells with intact membranes. And then finally, at the last step here, you'll run your controls and analyze using flow cytometry to quantify 4 different populations of target cells in each sample. So this includes cells in early apoptosis, which are not detectable by any other method. So these 4 populations that we'll be looking at include live target cells, early apoptotic cells, late apoptotic cells, and necrotic cells. So now we'll go ahead and analyze some flow data here for both the basic and the total cytotoxicity tests. So you can really see the breakdown of these different cell populations of interest and how that displays in flow cytometry data. So on the left, I do have flow cytometer data for the basic cytotoxicity test. And here I've gone ahead and we've created a plot with CFSE and 7AAD. And all the target cells are initially stained green. So the entire population of target cells can be counted in the R2 and R1 quadrants. After the effector cells and red live dead stain were added, all the dead target cells fluoresced red, shifting them up the y-axis and clearly distinguishing them from the live target cells. So the dead target cells are in R2 and the live target cells are in R1. So the other 2 quadrants here in this graph, the live effector cells are in R4 and the dead effector cells are in R3. So if we're going to go ahead and use the total cytotoxicity test kit, we would be further analyzing the R1 and R2 quadrants from the basic to isolate the apoptotic cells. So the total cytotoxicity test plot at the right has been extrapolated from the R1 and R2 quadrants at the left. So once the green target cells from R1 and R2 have been gated, we derived a plot of polycaspase apoptosis reagent SR Flicka versus the live dead viability stain 7AAD in this right total cytotoxicity plot. And here we can now analyze our 4 different populations of cells, which are our live cells and 3 populations of cells in the death process. So you can see in quadrant 1 that there are the live cells stained green. Quadrant 2 has early apoptotic cells that are stained both green and orange. Quadrant 3 has late apoptotic cells that are stained green, red, and orange. And quadrant 4 has necrotic cells that are stained green and red. And I just want to point out that quadrant 2 is of particular interest as no other method, such as PI staining or other assays, can identify this additional population of cells. The overall takeaway here is that researchers can now include apoptotic cells in their analysis of cytotoxicity, which does lead to more accurate results. So now I'd like to go ahead and cover the final product of our presentation today, which is our Fliss product line. Now, Fliss reagents, or fluorescent labeled inhibitors of serine proteases, are cell-permeant fluorescent inhibitor probes used to quantitate serine protease activity in cultured cells. Our Fliss assays are available with green or red probes. Serine proteases are defined by the presence of a serine residue at the active center of the enzyme. And the most characterized enzymes of this type are trypsin and chymotrypsin. So all living cells do have a base level of chymotrypsin-like enzymatic activity, which will vary with their physiological state of the cell as well as by cell type. However, activated serine proteases play major roles in several different functions, including apoptosis, markers of tumor malignancy, diagnostic and prognostic indicators of breast carcinomas, and neck and head carcinomas as well. So serine protease activity is also altered in a variety of other cell-mediated diseases, which can be related to transplant rejection and infections as well. So for our Fliss kits, you'd simply add the reagent directly to the cell culture media, incubate, and wash. Because ICT's Fliss inhibitors are cell-permeant, they will efficiently diffuse in and out of all living cells. If there's an active chymotrypsin-like enzyme inside the cell, it will covalently bind with the Fliss inhibitor and retain the green or red fluorescent signal within the cell. Cells containing lower concentrations of chymotrypsin-like enzyme activity will retain a lower level of fluorescence compared to the cells containing higher concentrations of this effector enzyme component. There's no interference from proenzymes nor inactive forms of this enzyme in this particular assay. Now, if your treatment is activating chymotrypsin-like serine proteases, positive cells will fluoresce brighter than the normal baseline negative cells, allowing you to clearly differentiate these populations. So to tie all the way back to the beginning of this presentation, you can use these Fliss kits in conjunction with our Flicka kits, which is what we're looking at in this particular figure. So what was done here is that HL60 cells were treated with camptothecin and apoptosis inducer for 3 hours and then stained with our green FAM-Fliss serine protease inhibitors and our red SR Flicka polycaspase inhibitor for an hour. These cells were then washed and analyzed on a scanning laser cytometer. And in this flow cytometry data, we can see that cells with active serine proteases will stain green along the y-axis, and cells with active caspases stain red along the x-axis. We can then see from some of these microscopy images here that there is a wide range with cells that range from A, which are only green cells that are showing serine protease activity, B, C, and D, which are dually stained cells showing both serine protease activity and caspase activity, and then finally E, which are only stained red for caspase activity. So with that, that wraps up the variety of fluorescent probes I wanted to talk about today. We went through a lot, so to recap, we talked through Flicka for detection of active caspases, Magic Red for detection of caspases and cathepsins in real time, MightOPT to assess mitochondrial permeability and health, cytotoxicity tests, and Fliss for serine protease detection. Since we covered a lot of ground, I did want to point out the variety of resources that we provide from ICT. We recently began our first ICT webinar series, so we do have our first webinar recording available on our website, and you can register for our upcoming webinars there as well. We'll also have some nice videos coming soon to show these kits in action and really show you how easy it is to run through these assay protocols. So I'd check back soon for those videos that should be up shortly. We update our blog with helpful articles, tips, and tricks for all things immunoassay related. And finally, we also have an extensive variety of technical documents that are hosted on our website for each of these kits to help you and support your work. This is a nice picture of our team here at ICT. Our team takes great pride in excellent customer and technical support. We offer very fast shipping, and if you have any questions about our products, you can often talk directly with the inventor of the product to help troubleshoot your process. For our international customers, we do have a large established global distributor network to assist you in getting you our products as fast as possible. And we also do have a guarantee on all our products, so if they don't work, we're happy to refund your purchase there. So that's about all the content I have here today. My contact information is as follows if you'd like to reach out to me with any questions or comments. Otherwise, I can take some questions at this time.
Thank you very much, Jackie, for that excellent presentation, bringing all that information to us. Before we get started on audience questions, I just want to remind our participants how they can submit their questions. Questions for Jackie can be submitted through the green Q&A button at the lower left of the presentation window. We'll try to get to as many questions as we can. I know it always takes just a minute for people to get their questions entered, so I'm going to give our audience time to do that right now. Again, as you can see on the slide, there's Jackie's contact information if you need to reach out to her. In addition, as Jackie mentioned, the slides will be available as a PDF download for any resources that you might need. So let's just give our audience just another minute for submitting questions online. Okay, I don't, do we have something? I don't see anything in our queue. I'm going to just turn it back over to Jackie for a minute with any final comments, words of advice, or any other information she has for our audience.
Thanks, Brenda. Yeah, just to wrap up, thank you again so much for joining us for our presentation. I hope you found the content helpful. If you do have any questions on any of our product lines, whether it's the fluorescent probes we discussed today, our ELISA solutions, or any of our custom services, please don't hesitate to contact me, and we can get all your questions answered. Thanks again for joining us, and I'll pass it back over to Brenda to wrap up.
Thank you again, Jackie, as well as our sponsor, ImmunoChemistry Technologies, for bringing this webcast today and making it possible. As I said before, slides are available as a PDF. There's something that you didn't catch. This webcast will also be available for on-demand viewing through March of 2016. You'll receive an email from LabRoots alerting you when the webcast is available for replay, and we would invite you to forward that email to any colleagues who might have missed today's live event. Thank you again for logging in and participating in today's webcast. See you next time.